The theory of panspermia suggests that life originated elsewhere and was transferred to Earth by interplanetary transport, specifically by the transport of microbes. This theory relies on the assumption that life can thrive in a foreign environment as long as conditions are favorable. An early example of this theory is the findings of the SNC meteorites. These meteorites are Martian meteorites and thus originated from Mars, and these meteorites were tested and were found to have contained gases that match the actual Martian atmosphere. Because these meteorites have gases from the Martian atmosphere, as well as survived the trip to Earth, this suggests that it's possible that other living organisms that were inside of these rocks could have survived the trip as well. Many experiments were conducted in order to test if these microbes could survive the different challenges that they would face during the 3 steps of interplanetary transfer. These 3 steps are ejection from the donor's planet surface, the donor being Mars. Number 2 is transit through space, and the 3rd step is entry and deposition onto the recipient planet, which in this case would be Earth. It is very likely that these microbes could survive these challenges that they have faced through their transplant onto Earth from Mars, because samples of bacteria have been made on the SNC and other Martian meteorites that indicate presence of Bacillus subtilis endospores, which are resistant to a number of challenges, including heat, UV radiation, desiccation, vacuum, lysozymes, and chemicals. The first experiment that was conducted was a ballistics experiment. This experiment was conducted by the researcher shooting a pellet into a target and then measuring the acceleration and jerk speeds, as well as the number of shots and the average % survival of these bacterial cultures. The conclusion was that the acceleration and the jerk were not an impediment, and the recovered samples were nearly at 100%. Another experiment was done under NASA with a vertical gas gun. This experiment tested spallation. The challenges present under spallation are shock and heating, and this experiment tested both of those in the absence of acceleration. The findings were that heating and shock were actually much more important to the ejection. The microbes tolerated the jerk and acceleration much better than the heat and shock, and the survivors for heat and shock were actually much less, by many factors of 10. The painting that is presented before you is meant to be symbolic and represent the first step of interplanetary transport. The different colors represent the different challenges that these microbes face during this first step of interplanetary transfer. The orange represents heat. The white strokes represent the acceleration. The white center represents spallation. The gray streaks represent the jerk, and the blue stripes represent the shock. The orange seems to consume the white because heat is more of an important factor during this first step. The blue streaks representing the shock are also seen as a very large impediment for these microbes and are thus the last barrier represented in this painting. The importance of the shock also accounts for the size of these stripes compared to the other streaks in the painting. The second step of this planetary exchange is the transit through space. Challenges faced in this step are vacuum with extreme desiccation, radiation, temperature extremes, and time. The experiment that was to prove that microbes could withstand these challenges was called PROTECT. During this experiment, Bacillus subtilis spores were spread onto aluminum discs that were placed onto a spacecraft in order to resemble spacecraft contamination. Many things were found during this experiment, one of which was that genes of unknown function were found from space and Mars spores, as well as the fact that the space exposure actually caused more damage to the spores than the Mars exposure did. Because this project was only over a period of 18 months, if more time was allotted to the spacecraft with these spores, more incidents of desiccation would be likely to be found. This second painting is meant to resemble the second step in the planetary exchange. The black is meant to symbolize space. The yellow circles are meant to portray the stars found in space. And all the other elements are meant to symbolize the different challenges faced by microbes in the space transit. The very big blue circle with the check mark inside is meant to be a symbol for time. The gray spirals and red spirals are meant to show the extreme temperature changes, and the green dots are meant to resemble the radiation that can be present in space. The reason why the stars are present is to show that the presence of these stars has an effect on the temperature changes, being as it is warmer in the vicinity of these stars, whereas in other spaces it is severely cold. The final step is entry into the recipient planet. The two challenges that are faced within this step are heating as well as impact. the experiment that was done to prove that microbes could withstand these two challenges is missile launching and impact into Earth. This experiment's findings showed that heating and impact were also very survivable by microbes. This last painting is meant to demonstrate this final stage of entry into the recipient planet. The red is again meant to show the heat, the effects of heat. The white is meant to show the impact created by the meteorite once it hits Earth.